All right, welcome back, everybody, to the deep dive. It's amazing to me how scientists are always finding ways to kind of hack biology to achieve these big industrial goals.
Oh, yeah.
And today we're going to dive deep into one specific example. We've got some fascinating research on how they're making these tiny organisms like microbes and bacteria and stuff resistant to certain things,
Yeah.
roadblocks basically. And that helps them make valuable products. So our mission, if you will, is to really break down how this resistance works
Right.
and why it's so important for the future of industrial biotechnology. This is stuff you're going to want to know. So what exactly are we talking about when we say making these microbes resistant?
Well, the idea is to kind of engineer or select microbial strains
Yeah.
that can ignore specific signals or substances that would normally inhibit them or even shut them down completely.
So they can just keep on chugging.
Exactly. And by doing this, we can, enhance their ability to make these specific compounds that we want.
Okay, that makes sense. So let's break this down a little bit. I think the first strategy we're going to talk about is creating these enzyme mutants that are insensitive to inhibitors.
Right.
Basically, I guess, finding or creating mutants that can keep doing their jobs even when there's something trying to stop them.
That's the gist of it. It's like
Yeah.
imagine a factory where a machine just keeps running smoothly,
Uh-huh.
even if somebody throws a little sand in the gears,
Oh, okay.
it just keeps going.
Gotcha.
And a really good example of this is a fungus called Aspergillus niger and its resistance to fluoracetate.
Fluoracetate. That sounds kind of intense.
Yeah, it's kind of mouthful.
What's the connection there? Why are we talking about this?
Well, the fascinating thing here is when Aspergillus niger, this fungus, develops a resistance to fluoracetate, uh, it starts to overproduce citric acid, like a lot of citric acid.
Okay, so how does that happen?
Well, the mechanism behind this involves changes in the aconitase gene. Now, the aconitase gene is what provides the instructions for making the aconitase enzyme.
Right, right.
And aconitase is this key player in what's called the citric acid cycle.
Okay.
And normally fluoracetate, it actually blocks aconitase.
Ah, okay.
It stops it from working.
So the mutations then in the aconitase gene make the enzyme less sensitive to that fluoracetate block.
Exactly. They kind of change the enzyme shape a little bit so that the fluoracetate can't bind as effectively.
Yeah.
And you know what that leads to?
I'm guessing more citric acid.
You got it. Because, with aconitase less active, the citric acid that's normally processed further down the line,
Mm-hmm.
it just starts to build up. Think of it like a bottleneck on an assembly line,
Oh, I see.
right? And citric acid is what's accumulating.
Yeah.
And you encounter citric acid constantly, by the way. I mean, it's in everything. Your fizzy drinks, your household
It's
cleaners.
true. It's everywhere.
It's really a great example of how we can, kind of tweak these biological systems for our own purposes.
That's really cool. And it's something that people, they interact with every day, but they don't realize, there's this whole kind of biological process behind
Yeah.
it. All right. So another example we have here is E. coli and methotrexate.
Yes.
I feel like I've heard that before. Isn't that a medicine or something?
Yeah, you're right. Methotrexate, it's used to treat cancer.
Oh, okay.
And it works by inhibiting an enzyme called dihydrofolate reductase. We call it DHFR for short.
Okay.
Now, DHFR is essential for making these building blocks that are needed for DNA synthesis. So basically, it stops the cancer cells from multiplying.
Okay.
But in E. coli, resistance to methotrexate can happen in a couple of ways. One is through having extra copies of the DHFR gene,
Yeah.
like suddenly having many copies of the recipe for DHFR.
So flooding the system.
Exactly.
Yeah.
Gene amplification, that's what we call it. Or there can be mutations in the folate gene. That's the gene that codes for DHFR.
So the recipe gets tweaked a little bit.
Yeah, and both of these things, they lead to an oversupply of DHFR.
I see. So either the cell makes so much of the enzyme that the inhibitor can't grab onto it all, or the enzyme itself is changed a little bit so the inhibitor can't bind.
Exactly. And while in bacteria, this leads to more of these tetrahydrofolate derivatives. The idea is similar to how cancer cells can become resistant to methotrexate in humans.
Oh, wow. So it's the same principles at play, just in different organisms.
Precisely. It's fascinating how these biological processes are so interconnected.
It is. It is. Okay, so let's move on to our second strategy. This is repressor-insensitive mutants.
Right.
So instead of fighting against an inhibitor, here the goal is to kind of sidestep the normal off switches for production.
Yeah, you got it. We're talking about mutants where the production of something isn't effectively turned off, even when it should be.
Okay, so example number one here, E. coli resistant to catabolite repression. Now that sounds complicated. What's the basic idea here?
Well, catabolite repression is it's this common way for bacteria to prioritize their food sources. So like if there's a preferred sugar around, like glucose, they'll often ignore other sugars like lactose.
Okay, picky eaters.
Nay, you could say that. But these E. coli mutants, often they have changes in genes like CROP or SIA,
Uh-huh.
or even in the lact promoter itself, and they become less sensitive to this glucose-driven shutdown.
So even if glucose is there, they can still use lactose efficiently.
Yeah, that's right. They can use both.
Okay, so industrially speaking, why is that a good thing?
Well, this is really helpful in situations where you might have a mixture of different sugars, right?
Mm-hmm.
Because these resistant strains, they can use all the available sugar more effectively,
Makes sense.
which leads to better overall yields and, a more efficient use of resources.
So it's like a workhorse that can eat anything you feed it.
Exactly. They're not picky eaters anymore.
All right, that's awesome. So our final example here is Bacillus subtilis and its tryptophan operon. What's going on here?
Okay. So the tryptophan operon is a classic example of what we call an operon. It's a set of genes that are all controlled together.
Okay.
Now, in this system, the presence of tryptophan itself actually acts as a repressor. So it turns off the production of more tryptophan.
Oh, interesting.
It's a feedback loop.
Makes sense.
But in Bacillus subtilis mutants with a deregulated tryptophan operon, this self-regulation is messed up.
So they just keep making tryptophan.
Yeah, pretty much. Even when there's already plenty available.
And that's because of mutations in certain genes, right?
Right. Often there are mutations in the TREP-ER gene. That's the gene that codes for the repressor protein. Or the TREP operator region. That's where the repressor binds.
Okay, so they either change the repressor or the place where it binds.
Exactly. And this leads to the overproduction of L-tryptophan, which is an essential amino acid. It's used in dietary supplements and animal feed.
Oh, wow. It's like super valuable then.
It is. And by kind of like breaking this natural feedback loop, we can generate a lot of it.
It's amazing how, scientists can just like figure out these little tweaks and then all of a sudden you've got like tons of this valuable stuff.
Yeah, it's pretty amazing.
So just to recap here, we talked about some really clever ways that scientists can kind of trick microorganisms into overproducing these valuable substances.
Yeah.
Or repressors, the things that would normally stop production altogether.
And that's a key point. It's all about manipulating these natural mechanisms to our advantage.
Right, and it's a super powerful tool in industrial biotechnology. It can be used to boost the production of a whole bunch of different compounds.
Oh, yeah, tons of stuff, from citric acid that makes your food taste good to amino acids that are essential for animal nutrition.
It's really incredible stuff. So I want to leave you with this thought. Beyond resistance, what other fundamental control mechanisms could scientists manipulate? Like what other internal switches or dials could we tweak to achieve even more specific goals?
That's a great question. I mean, the possibilities are really endless.
Yeah, it's an exciting field. All right, well, that's all the time we have for today, folks. Thanks for diving deep with us.
Thanks for having me.
And we'll see you next time.